It says, the following question has two parts. First answer part A, and then you're going to go on to part B. Let me zoom out a little bit. It says, a student in a biology class conducted an experiment to determine the effect of temperature on the rate of photosynthesis in a plant. They place an Elodea plant. Does it matter that you know what an Elodea plant is? No. I can tell you it's an aquatic plant. The plants they have in fish tanks. You see them in ponds. They're not algae, they actually are plants. Elodea plant into a test tube filled with water. Then the students place a test tube under the light and slowly increase the temperature of water. They record their results and display them on the graph as shown. Here's my suggestion to you. When you see a question like that, you should draw it. It'll help you. It might help you. It might not be helpful. Maybe it's just redundant. But if you go ahead and do the drawing and just draw a test tube, sketch it, you don't have to go crazy. Draw the leaves on a plant so you know that this is all underwater, right? And it says you increase the temperature. So you see how I drew it? I summarized what I know. Write down what you know. And sometimes that means drawing it. So then you have photosynthesis. And I'm going to go ahead and draw the graph here because it's easy to sketch. Here I have rate of photosynthesis. Here's the rate of photosynthesis. And here's the temperature. And it starts at 0 here on both. Obviously, it's increasing in this direction, it's increasing in that direction. Then you get this graph that looks something like this. And then you have different sections labeled. You have this section's labeled L, and then you have this section's labeled K. And here you have to know a little math, right? A little how to read a graph. Sounds like it's funny, but it's not, because it's hard to read a graph sometimes. And 25 degrees. This is degrees Celsius. And then the last section is here labeled N. So then they say, here it is. What does this graph show? The statement, which statement is supported by this graph? This is a graph of the data, right? They increased the temperature, they measured the rate of photosynthesis. How would you measure the rate of photosynthesis? What would you measure? How could you measure the amount of the rate of photosynthesis? How fast the plant grow could be a rate of photosynthesis. You're right, it would be a kind of an indirect way of measuring it. But the problem is that it takes a long time to measure that, right? It would take weeks and weeks and weeks. What's a faster way of doing it? Yeah. What would you say? Oxygen levels. You can measure the oxygen levels. The concentration of oxygen. What's another way you can measure? What's something else you can measure if you're not measuring oxygen? The concentration of what? CO2. Because it's going to take out CO2. If the more photosynthesis, the less CO2, the more oxygen. So those are two things you can easily measure. But I love your answers. They have show a good understanding of photosynthesis. All right, so now, the rate of oxygen production is lowest in M. So you'd expect M to have it's the rate of photosynthesis. Would you expect the rate of photosynthesis to be lowest at M? Rate of photosynthesis, M is the lowest on this chart? No, that's just reading a chart. That's not anything complicated. It's not. It seems like a trick question. But it's not. It's really easy. B, so it's not A. So B, the rate of oxygen production is greatest at M. Is this where you produce the most amount of oxygen at M? Sure. Your rate of photosynthesis is fastest, so you're producing the most amount of oxygen. If you know what photosynthesis is, you know M is the. But maybe maybe that's not the best answer. Let's keep looking. The production of water molecules is the greatest at L. Are you producing water molecules in photosynthesis? No, you're using them up. You have to know your equation, right? What's your equation? Water, and I just did it a minute ago, plus what? CO2 yields what? Sugar plus what? Oxygen gas. So if you're doing this more and more and more, right, then you'd expect less and less CO2, more and more oxygen, more sugar, and less water. Not more water. Don't put anything away. I don't know why you're putting stuff away. So now, the rate of the production of water molecules is greatest at L. Are you producing water? No, it's not. So that can't be the answer. The number of carbon dioxide molecules in a beaker is greatest at K. Greatest CO2 at K. Where's the greatest CO2? L. This is where you had the most CO2 because that's the beginning. As you go up, as you increase photosynthesis, what are you doing to the CO2 in here? It's decreasing, you're using it up. So obviously our M answer was right, and it is A. I feel good about this question. Part B, which statement explains the effect of temperature on the rate of photosynthesis? What do you see the rate of photosynthesis? How would you explain it? Somebody. How would you explain the rate of photosynthesis to change over time here?
So, the warmer it is, the higher the rate of photosynthesis.
That's right, until you reach,
25 degrees Celsius.
and then it just crashes. And the reason that is there's enzymes that are involved, and as increase the temperature of the water, you increase the speed of the reaction until you reach a point where the protein denatures, the enzyme denatures, it unravels. It loses its shape and therefore loses its function. And as soon as the enzymes stop functioning, you don't have photosynthesis anymore. All right, so oxygen molecules break down at temperatures above 25 degrees. No, that's not true, otherwise we'd all be dead. There would be no oxygen in the air. The rate of photosynthesis is unaffected by the temperature of water? No, we know it is affected. The number of chloroplasts in the cells decreases. Do you know anything about chloroplasts from this graph? No. Enzymes involved with the plant of photosynthesis are disrupted by temperatures above 25. That's the only reasonable answer. All right, have a good one. Well, the thought is this. You have to, in order to answer the whys and what fors, you have to think about what's happening, right? What we know of photosynthesis, what do we know about photosynthesis? There's these things called enzymes, right? These enzymes. They run biological chemical reactions, right? Biochemical reactions in bodies, in organisms. That's what makes them work, right? Remember that? And these things are proteins. And what do we know about proteins? They have shapes, right? Because remember that structure, right, is related to what? That's right. And we looked at cell membrane proteins. Remember that? We made those little channel proteins, and we looked at it has to have that specific shape for it to work. Well, what happens when you increase temperature? There's two things going on. If you're going A plus B yields C, and you have an enzyme with a specific shape that's working, making it happen. As you increase temperature, that's one way to make the A and B find each other in a flask, right? Or in a test tube. A and B have to hit each other. So they have to hit each other in just a certain way. That's how chemical reactions work. And so as you increase temperature, these move around faster. As they move around faster randomly, what do you increase the chances of? A and B finding each other. Does that make sense? Another way to increase that, how fast these two find each other, is using an enzyme. So the enzyme literally takes A, takes B, and puts them together. You remember our sodium potassium pump, where it would collect sodium and move it to change its shape and move it up, pump it out, and then it would collect the potassium, change its shape, and move it the other way. So just like that, it would A and B have to be captured by that enzyme, and as it captures, it allows A and B to become C. All right. So that's how enzymes work. But what happens when you increase the temperature too much? The temperature still increases this reaction. This reaction is still increased by temperature. It will continue to. It'll keep increasing as the motion of the atoms or molecules move around, they find each other more often, they have the chemical reaction happen. But what happens to the protein when it gets too hot is the hydrogen bonds break. And if the hydrogen bonds break, then the protein, what do you think happens to that protein? It unravels. So the amino acid sequence usually stays the same, but it loses its shape. So it doesn't look like Pac-Man anymore. It's just a blob of protein. Just like the DNA doesn't look like DNA anymore. It looks like that slime that we had in that test tube when we isolated DNA from strawberries. It didn't look like a double helix, right? It looked like just a snot, right? And when you look at it, what happens to the protein when you overheat it, when it heats up too much, not cook it. If you heat it up too much, you cooked it. You permanently changed it. You've changed the covalent bonds. Here we're talking about heating it up enough to break the hydrogen bonds so you lose the shape. If you lose shape, you lose function. Is that clear? So we call this word, well, we have a word for this, it's called denature. We call it denature. You get rid of the natural shape of the protein. You denature. When a protein can be denatured, you let it cool down, and guess what happens? It goes back to its original shape. So what happens when you heat up a protein too much, but not so much that you cook it, right? But just enough so that it changes, it loses its shape, it stops functioning. And that's what happened here, you see. It got to this point, and it was heated up too much, and the enzymes don't work anymore, and the chemical reaction stopped. If you cool it off, they start again. And if you remember, we called this homeostasis, right? Where we said that, I think the example I used in class, it was a few months ago, it's like back in, I think, November. If you go to the hospital, you have 105 degree temperature, 110, right? Your enzymes are unraveling. They're not working anymore. Your proteins in your cell membranes could even start to break down. You're starting to cook, right, after a certain point, but this is before you cook. This is before you affect the covalent bonds. The proteins are unraveling. And because the hydrogen bonds are not working, the proteins are losing their shape, they're losing their function. If they lose their function enough, you die. So you go in the hospital room, you go in the emergency room, and you're too hot, you're 110, you have a really bad fever. What are you going to do? And by the way, this is the way your body fights bacteria. Is when it has an invader, it turns up the heat, and what happens to the proteins in the bacteria? They unravel, and what happens to the bacteria? They die. So they're more sensitive to these temperature changes than we are, so that's what our bodies use to kill them. But if our temperature gets out of control, it gets so hot that our proteins start to unravel, then we got problems. We begin to die. So you're in an emergency room, or your child is in the emergency room and has this extremely high fever. What are they gonna do? What's the first thing they do? What do you think they do?
IV.
Huh?
IV.
No, IV is not a bad idea, but that's not the first thing they do. They'll probably give you an IV of antibiotics, yes. So what are they gonna do before that? Before they even do that, because the antibiotics will kill the bacteria. That's good, but you're dying because you're too hot. What are you gonna do? Cool them down. They literally put you in an ice bath. They literally have a bathtub full of ice that they put you in, just to bring your temperature down so your proteins can renature, gain their shape back again. So denaturing is losing their shape, and renaturing is gaining their shape. And by the way, that's one way we can stop proteins from working in the laboratory. We have this thing called PCR, this polymerase chain reaction. That's an enzyme that makes DNA, copies DNA, helps copy DNA. That's what polymerase, remember DNA polymerase? Well, we have this special enzyme that is isolated from a bacteria that can work at a higher temperature. But we can increase the temperature so that only that enzyme is working and it's doing its job. Then we increase it and we can shut it down. We can shut off that enzyme and turn on with that temperature change. because we can renature and denature that enzyme. We can open up the DNA molecule, close it up by breaking the hydrogen bonds using temperature. So we can use this really basic function of life that proteins denature and renature back and forth depending on temperature. We can use that in a laboratory for experiments. So you may hear this again in case the kids that are going to get this through YouTube, they might want to hear an explanation again. So you might hear it again tomorrow. All right. I would like you guys to explain it tomorrow. Well, maybe they won't. It won't be tomorrow, will it? It'll be Friday. Yeah. So I want to have them for 2 days. So hopefully they'll watch this video. So let's go back to this question. Take a look at this chart. This is the chart that you have to answer. You're going to give me the answer. Are you ready? You can tell me which one of these is right. Tell me which one's wrong. Here we go. Which statement is supported by the data in the graph? So this is the data in the graph, right? Which statement supports that? Are you ready? The rate of oxygen production is lowest in section M. Yes or no? Wrong. Why is it wrong? It has the highest rate of photosynthesis, and you should be producing the oxygen the most, not the least, right? So that's why that's wrong. So I'm literally going through, as I'm doing this, by the way, and you guys should be thinking about doing this too, is use the tools on the exam that they give you. I'm striking through the ones that I know are wrong, so that when I'm looking at these questions, I can honestly say, okay, it's between these two. Because I already struck out the other ones, I'm for sure. I'm not going to go back and don't strike them out unless you're sure it's wrong. So make a commitment to yourself. Trust yourself. All right? If you're not trusting yourself, it's going to be hard for you to get as good a grade as you can get. Do not change an answer unless you know for sure you made a mistake. It's like, oh my God, that was a stupid mistake. Let me change it. Okay. As long as you're sure. But if you're just like going back and forth and thinking it's A or B, A or B, I chose A, but I think it's B, I'm just going to go with B. Don't do that. Stick with the original. Your first choice is usually your best. Unless we all make mistakes. Unless you make a silly mistake, then go back and change it. But make sure you're aware of why you're doing that. All right, which statement is supported by the graph? Data in the graph, the rate of oxygen production is the greatest in M. I agree with that, but are we just going to choose B and then move on? No, we're going to read all of them. The production of water molecules is greatest in section L.
It doesn't talk about water.
Now here's a better question. Does photosynthesis produce water? No, it uses water, right? That's why plants need water. Cellular respiration does produce water, but this is photosynthesis. That's what we're measuring here. So that's wrong in several different ways, right? Multiple ways. The number of carbon dioxide molecules in the beaker is greatest in K. No. You would think, see, I understand, but look at the graph. You'd think it's greatest, the concentration of CO2 is greatest in K. I agree with you that they're greater in K than in M. They're greater in K than in N. But where is CO2 in the test tube greater even than K? L. Because it's earlier, right? As you go along the line, you have less and less CO2 available. You're right. I know where you're going with that. You're absolutely right. But L is where the concentration of CO2 is the highest. But let's say you confuse yourself and you're like, wait a minute, I think it might be true. It might be K. But then how do you justify that the rate of oxygen production is greatest in M is for sure, right? Which one would you choose? You would choose B, right? Because it is M. For sure, M is the highest. So you've got to go with the one that you're for sure. If you have two answers and you're just like, maybe this one's right, maybe this one's wrong, but this one's definitely right, then choose the one that's definitely right, right? Choose your best. This is multiple choice. This is the best answer. All right, let's go with B. Which statement explains the effect of temperature on the rate of photosynthesis? Oxygen molecules break down at temperatures above 25 degrees Celsius. It doesn't make, I know that everybody's asking, does the chart match? But listen to the statement. Oxygen molecules Break down. Well, what is it? What's an oxygen molecule? Let's remember what it is. An oxygen molecule is an oxygen double bonded to an oxygen. That's an oxygen bond. These are covalent bonds. O2, this is what we breathe. Does an oxygen molecule break up above 25 degrees Celsius? On a hot day, does oxygen start to break apart? No, otherwise we'd be dead, right? We wouldn't have any oxygen to breathe. On a hot summer day, there wouldn't be any oxygen. It takes a lot to break this down, right? Or to build it. We build it, and then we use it. In cellular respiration, we use oxygen to make water, but it takes a lot. Remember all the steps that it took to get that energy in a position to be able to do that. It does not happen spontaneously. You can break oxygen molecules apart with temperature, but it's much, much higher than 25 degrees, right? So that one's wrong. So let me get this strike through so when we look at it later, we're on the same page. Oh, this thing is sensitive. All right, so the rate of photosynthesis is unaffected by the temperature of water. The temperature of water, the rate of photosynthesis is not affected. What would it look like if the rate of photosynthesis was not impacted by temperature? It would be a straight line. And do we have a straight line? No, we don't. So that's not an answer. So even if you were questioning A, B is certainly not the answer. You could always put a question on A, right? Like, I don't know, I'm confused at what that means. Okay, we'll put a question mark on it. But don't choose it until you're sure it's the best answer. The number of chloroplasts in a cell decreases as temperature increases. So as it gets hotter, you start to what? Kill off chloroplasts? It doesn't work that way. It doesn't work that way. Number of chloroplasts in the cell are determined by things not to do with temperature. Enzymes involved with the plant photosynthesis are disrupted at temperatures above 25 degrees. And that makes sense. That one's the one that makes sense. So if you, for some reason, were questioning this idea of what's going on with that one question of what happens to oxygen above 25 degrees, you know that the enzymes have to stop working at 25 degrees because that's how these chemical reactions work. I think we've hit it pretty hard this year that enzymes drive everything that goes on in your body. And enzymes are the key. So if something stops working, it's because of an enzyme. 99% of the time. Why do people get diseases? Because you get a mutation in your DNA. But it's not the DNA that's the problem, is it? The mutation in DNA changes what? The amino acid sequence, which changes what? The protein, which is the enzyme, right? So you change the enzyme, then you got a disease. It doesn't work. Everything has to do with those enzymes and how they do their job. If we look at this, then we can see that we literally have chosen the right answers, and we've done it very efficiently. And when I'm done with this test, I can go back and review the answers and see clearly, right? And by the way, you should apply this for your math test today and tomorrow as well. Cross out the ones that you're sure are wrong. Because even if you have two left, you're improving your odds that you're going to do well. Exactly. So this is like test-taking skills, right? Write down what you know and try to figure out what you don't. Okay, a scientist is examining a pedigree that includes several generations of an organism with XY chromosome sex determination. What does that mean? Why would they do that? Why would they say that? Because they want you to think about an organism that determines sex, whether there's something's a male or a female, like humans do. Because that's how humans decide, right? Different organisms decide sex in different ways. When I say sex, I mean male or female, all right? So sex determination in this organism is XX/XY. They could have just told you in a human, but people don't like questions about humans. right? They get sensitive. So they say an organism that has this way of doing it. Which pattern of inheritance would support the hypothesis that the trait being studied is a recessive sex-linked trait found on the X chromosome? So before we go ahead and try to answer the questions, you want to go ahead and look at the questions, see what you think, make a choice, and then we'll go review the content. Before I ask you what you think your answer is, let's go ahead and take a look at what some of the background information about X-linked traits are. Remember that X-linked traits are found on chromosomes, the chromosome X. And the really special thing about the chromosomes X and Y is that the males have only one copy of that gene. Of all the genes on the X, the male only has one copy, where the female has two copies. That gives the female an advantage with sexually determined or sex-linked traits. And the advantage is for those traits that cause diseases, the female has a backup copy. Does that make sense? So, but the male then has a disadvantage because they only have one copy. And remember with most recessive traits, if you have a genetic recessive trait, you need two copies of that gene to see it, right? So you have little r, right? That's the only way you're going to see, let's say it's, I don't know, some disease X, right? I don't want to say a disease, so let's just say it's a disease. It kills you. You get little r, you're dead. But the chances of you getting little r are determined by your parents, obviously, but you need two copies of little r to get it. If you have even one big R, you're not dead. That's great news. But the problem with sex-linked traits is that if you're a male, you only get one copy. So let's take a look at an example. Big C is the dominant form of the gene, and it means you don't have the disease. If you have a little c, you have the disease. Two little c's, you have the disease. Or one little c, and if you don't have a big C, you have the disease, right? Does that make sense? So if we look at what happens with a male that doesn't have the disease and a female that does, so mom is going to give you a little c, but to his daughters, the male gives protection because he has an X that does not give his daughters the disease. It actually protects against the disease. But to the male to his sons give him a Y. And because his sons have Ys, the only X that the boys have is that recessive disease-causing gene. So the boys end up with that disease. These are X-linked traits. There are a lot of X-linked traits. And these diseases are what you see in the population when you do a pedigree is you see more boys have it than girls. And that's really going to be key. So now let's go back to the multiple choice. It says, which pattern of inheritance would support the hypothesis that the trait being studied is a recessive X-linked trait found in the X chromosome? The trait is only expressed in males who have a father with the trait. Yeah, that doesn't make sense, right? The father gives a Y, it doesn't give an X. Does that make sense? Now, what my problem is, here's my problem with this idea of sex-linked, because there are Y-linked diseases, and that's sex-linked too. So they really should have said X-linked if they wanted to do that. So, instead of crossing this one out, I'm going to put a question mark till I get to the other answers. So you got to be careful with that, right? Because if it's a Y-linked trait, then this is possible. They say what? Oh yeah, I'm sorry. They did tell you it was X-linked. I forgot that. I was skipping back and forth. I didn't see the last part. They say it's found on the X chromosome. It's X-linked that's found on the X chromosome. So usually it's phrasing. So okay, yeah, they tell you it's on the X chromosome. So that's important information that I missed. Sorry. So A is wrong, right? So I'm confident that A is wrong because Dad does not give them, his boys, an X. He gives them a Y. So that information is not very relevant. The trait is expressed in half the female organisms and all the male organisms. Well, that's possible because more males than females have it. Is it possible for a female to get it, to a recessive gene? Not from a father, not if the father has a dominant form, but if the father has the trait and the mother has a trait, then yes. In fact, all the females will have it. So it's a matter of what's going on. In fact, if we go back, let me switch back and show you a couple other possibilities. So we know it's X-linked, so let's take a look at another possibility. What if Dad had it, right? But Mom, and by saying Mom, it could be some organ, it could be a plant for the intents of this exercise. But let's say Mom only has one copy. Mom only has one copy that's recessive, and the other one's dominant. So at this point, if Dad has it, then what are the odds that a girl's going to have it? One of his daughters is going to have it. What are the odds that the girls are going to have it? Anybody? I feel like Ferris Bueller's teacher right now. What are the odds that this mating is going to produce a female with the disease? A female. What percentage of the females have the disease? Let me rephrase it that way. What percentage of the females have the disease? These are the females. 50%, that's right. And 50% of the boys. 50% of the boys. So it is possible for a female to get the disease. It's just more likely that the boys have it. Does that make sense? And remember, in a pedigree is not one mating, right? So pedigree, you're looking at many generations and many matings and many different genetic combinations. So if you see this, and when you look at the overall situation, out of 8 different possibilities, 1, 2, 3 males have it out of 8. One female has it out of 8. So you see, most of the time, the males are going to have it more often than the females. All right, so don't forget that even though in this situation it's 50-50 for both, when you're talking about a pedigree, you're talking about a big sample. All right, let's switch back for a second and finish this problem. The trait is mostly expressed in females who have a paternal grandmother with the trait. What does that mean? A paternal grandmother with the trait. Anybody want to tell me what that means? There's only 4 or 5 of us, man. We should be able all to participate in this. Think about it. Just make a statement. Be wrong for a second. The trait is mostly expressed in females who have a paternal What does paternal mean? Say that again? From the father. So let's take a look at what that means. And again, if you're in a problem and you're like, what do they mean by this? I would go ahead and draw it out. So they're saying that the paternal this is the father, right? There's dad. So the paternal grandmother has the trait, right? So that means she gave him a what? What did she give him? This recessive, so what did she have to give him? What did she have to give him? Why is there mumbling? If she has, what does she have? She has two X's with what? What is her genotype? Sex-linked recessive. It has to be small, right? She has to be recessive, both of them. So what does she give him? The recessive trait. But when you're talking again about a pedigree, does the dad determine what his females have? The dad can protect the female, but if mom doesn't have it, what is she going to give her daughters? The dominant. Does that make sense? So even though dad will give his daughter that X chromosome with the recessive gene, their mom could give them an X chromosome with the dominant gene. Does that make sense? So really, that idea that the trait is somehow that most of the females, mostly expressed in females, is not correct. All right, and I'm telling you what the rule is, and you saw me write it down. More boys than girls, always more boys than girls with X-linked traits. So that means that this one here is also wrong. So the only right answer is the traits expressed in half the female organisms of all the male and all the male organisms. That's because more boys than girls are going to express the gene. Okay? All the rest of these were ways of confusing you. They were trying to say, does this kid know what they're talking about or not? If they know what they're talking about, they know B is true. So if they're confused, even if they're confused, which one are you going to choose? The one you know is true or the ones that are confusing you? The one you know is true. Go with the one you know is true. Okay, this is an easier one. I don't think, maybe I should draw it out. Maybe I should. I think I will draw it out just because I'm sending this home to people. And we have some time. So I know you guys have to leave. Are kids coming in here then? I should pause. So this is question 13 on the practice, and what they give you is a cell membrane. Obviously, I think it's obvious. If it's not obvious to you, this is back to one of those things that you really need to think about. So you have a cell membrane, and then you have a bunch of dots. They don't tell you which side's high and which side's low.
You can't see them.
I know. You see the picture in front of you. Right, I'm just sketching out the picture on the iPad so that when we flip over to look at it, we can talk about it without wasting time. So we have then we have particles in time 2. You see the particles are on one side. Did they tell you which side's high and which side's low? No, they don't. They don't tell you anything about high and low, but they give you a picture. They expect you're going to do what? Bless you. You're going to figure it out. You're gonna compare them, and you're going to make an analysis of them. Okay. So they give you 3 times, and here's what I'm going to tell you to do with every one of these problems. When they give you an illustration or they give you a movie, look at the diagram first. Oh, wow. Let me pause this so I can deal with this. All right. So when we're looking at this, the first thing you do to an illustration is you look at the diagram and you figure out what's happening here. You're sitting down out of dress code after I just got done telling you to get in dress code. Now, I don't know what else to say. All right, so you look at the diagrams and you figure out what's going on first. before you even address the question. You do not want to try to figure out any answers or whatever. Just looking at, just thinking to yourself, what are they talking about here? Put your mind in the game. All right? Does everybody here follow football at all? Look at football, watch football? No? Anybody, what kind of games do you guys, are you something?
Fortnite. Yeah.
Wow, what a waste of time. Why am I asking a question? All right, enough. I'm sorry I wasted all our time by trying to come up with an analogy.
You just said basketball.
All right. So, when we're talking about the strategy of looking at the illustration first, what it is you want to look and see, put your mind in the game that's in front of you. Focus, start to bring ideas into your brain that you have covered. You've thought about it, you've done the practice problems, but now you want to look at this problem and already, what am I doing when I'm looking at this? I'm already thinking about osmosis. I'm thinking about diffusion. I'm thinking about high to low. I'm thinking about active transport being the opposite. I haven't even read any words yet, right? All I see is 3 pictures. I'm looking at the labels. I'm asking myself, do they say water? No, they say particles. So they use the word particles crossing the membrane. Then I ask myself, what am I looking at? I'm looking at something that's labeled time 1, time 2, and time 3. I'm looking at that. And I'm thinking, well, there must be the same membrane at 3 different times. That must be what's going on here. So anybody that already my brain is preparing to start to understand what the question is. Because a lot of you jump to trying to answer the question, bless you, and you end up answering questions that are not being asked of you. No one asked you about it, those whatever it is that you're answering, but you answered that question that wasn't asked, and you get the answer wrong, and you think you got it right, and you might have written down the right answer for the question that was in your head, but it's not the question you were asked. So that's why looking at this problem and looking at the picture and thinking about it first, before you go reading anything, helps you start to process what's going on. But you don't want to make any conclusions yet. Does that make sense? I'm thinking these ideas almost passively. I'm not committing to them because I don't know if it's osmosis or diffusion. I don't know if it's active or passive transport yet. But I'm moving I'm bringing these words and these ideas into my brain by looking at it. And I'm not spending as much time as I'm spending now with you talking about it. You're spending a few seconds, maybe 30 seconds looking at the picture trying to figure out what's going on. That's it. Then it says a diagram models in a time-lapse movement of particles across the cell membrane. The particles have characteristics that allow them to pass freely across the membrane. What did they tell you right there? That these particles pass through the membrane without being restricted. So that probably tells you, if you remember anything, that they're nonpolar, right? And these are good points. Let's write them down. Nonpolar, that's number 1. They're probably small. Someone said you're not using ATP. Why did she say that? She said that because obviously you start off with time 1 as this side being high and this side being low. In time 2, you see the particles are moving in this direction from high to low. Yeah. Let me switch it. I was going to switch it in a minute, but let me go ahead and switch it. So nonpolar, so that means again it's non-charged, it's nonpolar, so you can cross the membrane, it's small, it's moving from high to low, and it moves in high to low in both directions, and it's some movement in both directions, but it's mostly from high to low. And then you get some equilibrium where you're moving in both directions, but the movement in both directions is constant, right? So that's why this person here said no ATP is being used. Could you please sit down? We're going to finish this before we move on, before we switch classes. Go ahead and take a seat. Thank you. Waiting. Someone, Cameron, could you close the door, please? Thank you. So when we're talking about this, we know automatically all this information, and I haven't really even read the question yet, right? All this I got from what? This is what I know. This is problem solving 101. Write down what you know on scrap paper. Read what they're telling you. Look at the pictures, because the pictures give you a lot of information. The illustrations give you a lot of information. Write down what you know based on those illustrations, and hopefully that helps. Now we go to the question. Now we finally get to the question. It says, select all the statements that describe the particles or their movement based on time-lapse diagram. On the time-lapse diagram. So now, what does that suggest to you? That you're going to choose one?
No,
More than one. And maybe none of them.
Select one.
Maybe all of them. Maybe just a few of them. Right? So this is kind of crazy. Do you see why I gave you those difficult questions over the year that asked you to choose more than one of all that apply? Why did I do that? Because I know it's going to happen at the end of the year, at the end of the course exam. So number one, the particles crossing the membrane do not have a charge. Is that true or not? That's true. Why is that true? Non-polar means they don't have a charge. If you're confused about that, if you're surprised about that, write that down as one of the things you have to review before next week. This is how you identify what you don't understand. I'm telling you right now, I see 4 or 5 of you lay your heads down, not paying attention, looking out a window. Those are going to be the people that are confused on the test and don't do well, or not, at least not as well as they could have. If you're looking in there and you're saying, I do not have a charge, you chose the wrong answer, you're confused about it, that's something you have to really do some work on before next week. The movement across the membrane requires energy from ATP. That is wrong. We already identified that we're not going to need energy. Movement across the membrane requires notice how I'm using the strikeout function. Please use the strikeout function during your test. They will help you. The movement across the membrane requires energy from glucose. No, we said it didn't require energy. We already identified it was passive, right? It's diffusion. This is just diffusion. Non-polar, small. No, it's going from high to low. Movement across the membrane does not require added energy. Yes, that's true. The particles crossing the membrane could be proteins or carbohydrates. No, they cannot be, because proteins and carbohydrates are big and they're polar. So I said they were small. What do we know? I guess there's a lot of review that some of you have to do about what crosses the membrane and what doesn't. You better go back and review those videos. You better come in for tutoring. Whatever do you need? The particles crossing the membrane could be water or oxygen. Yes, they could be. Is water, even though water is polar, it's small, and oxygen is not polar and small, and we know some water does get through the membrane, so this could be water or oxygen. That's true. The particles crossing the membrane have a strong positive or negative charge. No, we already say they don't have a charge. They're non-polar. That's what non-polar means. So the 3 that we would choose are particles crossing the membrane do not have a charge. The movement across the membrane do not require any added energy. The particles crossing the membrane could be water or oxygen. Listen, based on what I see in this classroom, about 5 of you have some work to do before next week. Have a good one. Get to your class. If somebody needs a pass, tell them to text me.